Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive into this fascinating world of biological transformation, please take just a quick moment right now to like, subscribe, and share this deep dive with friends and family. Your support really does drive the SANS Science Experience and helps us bring you more of this cutting-edge science. Okay, so today we're unpacking one of nature's most, well, energetic spectacles, metamorphosis. If you think about it, this is maybe the single most energy-expensive full-scale developmental overhaul. I mean, it's experienced by over 3/4 of known animal species. We aren't talking about just slow growth here. We're talking about a really abrupt, massive developmental transition. It needs wholesale tissue remodeling. The energy budget for this must just be astronomical.
Oh, it absolutely is. And that's what makes the research we're focusing on today so critical, When an organism goes through this kind of profound change, the real question is, how does it manage the fuel supply? Specifically, how does the body shift its reliance between, say, aerobic metabolism, that's the steady oxygen-dependent fuel, and anaerobic metabolism, which is more about explosive short-burst capacity?
Right, and the researchers we're looking at used a classic model for this kind of transition, the fully aquatic amphibian Xenopus laevis, the African clawed frog.
Exactly. They investigated how energy production is managed across the entire life history, so from the larval stage, the tadpole, all the way through to the 2-year adult frog. And the central finding, which is just a great piece of biological precision, really, is that these metabolic shifts aren't happening organism-wide. They are highly organ-specific. Basically, each tissue gets its own custom metabolic toolkit.
That customization, that's the key, isn't it? That's what we're diving into. So they focus on 4 specific organs, which, well, during metamorphosis, they experience radically different fates. Let's maybe walk through their roles.
Sure. First, you've got the gut. This is kind of the demolition zone. It shrinks by about 70%, undergoes really rapid programmed cell death, that's the apoptosis, and then rebuilds a completely new intestine, one that's tailored for a carnivorous diet, replacing the tadpole's old herbivorous setup. So this needs maximum immediate energy just for that remodeling.
Okay, massive rebuild. Then we have the hind limb muscle. This is more like the construction site, yeah?
Exactly. It's either completely new growth or rapid transformation of existing muscle fibers, all geared towards how the frog will move after metamorphosis.
Yeah.
So this energy demand is more long-term, it's sustained, supporting mobility for the adult frog.
Got it. And finally, the 2 essential workhorses that just have to keep going through the whole thing, the liver and the heart.
Right. While their structural remodeling is less dramatic compared to the gut, their sustained functional demands increase quite dramatically. The liver has to manage all the metabolic chaos and detoxification, and the heart has to ensure constant, efficient oxygen transport to support this growing body and its new behaviors.
So, okay, 4 organs, 4 totally different energy needs profiles. And to measure how they handle this, the scientists use these enzymatic biomarkers. Think of these as kind of like probes, right? Telling us which parts of the body's energetic machinery are being built up or maybe torn down.
Exactly. So to measure the aerobic machinery, they basically asked 3 key questions. The first question was essentially, are we building more engines? And this is the question of mitochondrial volume.
Okay, the number of power plants.
Yeah.
And they measured this using citrate synthase, or CS, activity. CS kicks off the Krebs cycle, so its activity is a pretty good proxy for how much basic aerobic machinery is physically there.
Precisely. Then the second and third questions addressed efficiency. How efficiently are those engines running, and is the oxygen flow maximized? They measured oxidative capacity using succinate dehydrogenase, or SDH activity, that's complex 2 in the electron transport chain.
Complex 2, right. And finally, they measured oxygen consumption capacity via cytochrome C oxidase, or COX activity, which is complex 4, the enzyme doing the very last oxygen-dependent step of the whole chain.
So CS tells us the sheer volume of the power plant, you could say, while SDH and COX tell us more about how hard that plant is being worked and how efficiently it's actually burning oxygen.
Okay, so that's the full aerobic toolbox nailed down. Now, they also measured the other side, the high-burst oxygen-independent system, that's anaerobic capacity, using lactate dehydrogenase, or LDH, activity.
Yeah, the enzyme that's crucial for converting lactate, especially during periods of oxygen dead or intense activity.
Right. Okay, now we get to the data, and this is where the precision of this metabolic reprogramming really starts to shine. Let's look at the aerobic side first, starting with mitochondrial volume, that's CS activity, across the developmental stages. The contrasts are huge.
Just look at the difference between the gut and the hind limb. It's striking. The gut's mitochondrial volume actually increased notably during that core metamorphosis stage, from NF60 to NF66.
Which makes sense, right? It needs that immediate, intense aerobic fuel to power the huge cost of tissue destruction and remodeling that's happening right then and there.
Exactly. But then the hind limb muscle, which is building structures for long-term use, it showed a totally different strategy. Its mitochondrial volume showed a much more gradual, steady increase from metamorphosis until the frog was about 7 months old, and then it stabilized in the adult.
So it wasn't about a sudden burst of energy supply. It was about sustained, gradual buildup to support long-term growth and, well, hopping around.
The energy plans were clearly customized. And then you have the two organs that need to be consistent. The heart was remarkably stable. It maintained a pretty constant mitochondrial volume across every single developmental stage they measured.
That's the dependable engine, I guess. Can't afford to fluctuate too much.
Right, but the liver is the one that really bucks the trend here.
Yeah, what's going on there? It showed a consistent decrease in mitochondrial volume from metamorphosis all the way into the 2-year adult stage.
If the machinery itself is shrinking, what does that imply? Is it just getting way more efficient, or maybe doing less metabolic heavy lifting as growth stops?
Well, it probably suggests the latter, at least relative to its size. The intensity of its metabolic role likely peaks early on, handling that initial flood of hormones and structural materials during metamorphosis. And then it sort of settles into a less energetically demanding regulatory role later in life. Okay, that makes sense. So if we shift from volume CS to actual capacity SDH for oxidative efficiency and COX or oxygen consumption, we really see this focus on post-metamorphic performance, especially in the heart and the hind limb muscle.
Absolutely. Their oxidative capacity, the SDH measure, was actually lower during the metamorphic chaos, but then it increased sharply and stayed high in the mature frogs.
So that's a direct metabolic adjustment. It's linked to the increased function of adulthood. More swimming, better foraging, just sustained high activity levels.
Definitely. But then we look at the liver again, and it gives us that counterintuitive pattern.
Yeah, the decline. Its oxidative capacity, SDH, was highest right at the onset of metamorphosis, then it decreased later. Meanwhile, its oxygen consumption capacity, the COX measure, increased until about 7 months, but then saw a marked decrease in the fully mature 2-year adults.
Which kind of reinforces that idea that the liver's metabolic workload shifts quite dramatically over time. High initial oxidative capacity supports the huge chemical demands of metamorphosis itself, but that demand just falls off once the system stabilizes in the adult frog.
And the gut. What happened with its capacity?
The gut's oxygen consumption capacity, COX, was actually pretty stable across all the stages, suggesting that once the new gut is built, its functional energy demands are quite consistent.
Okay, that covers the long-haul, steady aerobic system. But a frog needs explosive power too, right? The ability to leap, maybe fight gravity if it's partially terrestrial, escape predators. That demands a completely different system. Let's look at the high-power, short-burst capacity. Anaerobic metabolism, measured by LDH activity.
Yeah, and what's fascinating here is that the temporal pattern for anaerobic capacity was remarkably consistent across almost all the organs they looked at. Basically, a gradual and consistent increase from the metamorphic stages right into adulthood.
So this looks like a clear adaptive response. It was particularly strong in the heart and the hind limb muscle, wasn't it?
Very much so. High anaerobic capacity means they're prepared to use glycolysis for rapid energy bursts, which means generating high levels of lactate during intense activities like jumping or swimming fast.
Okay, but you said almost all organs. There was an exception.
There was. One tissue resisted this general trend towards increased explosive capacity: the gut. The gut was the only organ where anaerobic capacity actually decreased during metamorphosis.
Huh. Why would the gut need less capacity for oxygen-independent energy? That seems odd.
Well, the source material suggests this might be linked to the shifting microenvironment inside the gut itself, the gut lumen. As the tadpole gut gets replaced by the frog gut, the whole community of gut microbes changes completely.
Ah, the microbiota.
Exactly. And those microbial shifts can actually alter local oxygen levels within the gut, which could in turn influence which energy pathways the surrounding gut tissue needs to prioritize or express. It's like a localized biochemical shift influencing the metabolic programming.
Interesting connection. Now let's try and connect the dots on that high LDH found across the adult organs, particularly the muscle, heart, and liver. You said it points to high lactate production, but also maybe usage. Can we trace like an energy recycling cycle here?
Yes, I think we can. It paints a pretty sophisticated picture. So when the adult frog jumps aggressively, Its hind limb muscle produces a high level of lactate through glycolysis. Quick energy.
Right, the burn.
Yeah.
And lactate is often thought of as just waste, but it's not, is it?
Not at all. It's recoverable energy, and the high LDH activity in the heart suggests it plays a major role in recovering this energy. The heart is highly specialized, and it often contributes to greater lactate consumption, meaning it preferentially takes up that exogenous lactate coming from the blood, the stuff the muscle produced, and uses it as a highly efficient fuel source for its own pumping.
Okay, so the heart is literally consuming the muscle's spent fuel. Clever. And where does the rest of the lactate go?
To the liver. The liver acts as the body's metabolic refinery in this scenario. The high anaerobic capacity there and the subsequent high transport of lactate to the liver allows it to convert that lactate back into fresh fuel glucose through gluconeogenesis.
Making new glucose from non-carb sources. That's
Yeah.
the key piece. So the liver takes the spent fuel, processes it, and then recirculates the resulting glucose back to the muscles for more activity. It completes this really elegant cycle that maximizes performance during those high-intensity moments.
That's it. And this, I think, is the true aha moment of the study. Metabolic flexibility and this organ-specific metabolic reprogramming. They aren't just minor tweaks. They are critical, precisely orchestrated components of these major life history transitions.
So the organism isn't just flicking a single switch for its whole metabolism, it's applying surgical precision. The energy demands for intense tissue remodeling in the gut are met by one specific metabolic plan, while the energy demands for sustained locomotion and recovery in the hind limb are met by an entirely different specialized plan.
Exactly. And this concept of customized metabolic programming is likely incredibly relevant way beyond Xenopus. It strongly suggests that for pretty much any species undergoing metamorphosis, whether it's an insect, a fish, another amphibian, metabolic flexibility is probably the core strategy. It's what optimizes performance when the animal transitions into a completely new ecological context, a new way of life.
It really makes you wonder then, does this whole system of customized metabolic shifts become even more complex when the transition isn't just a structural change, but a shift from water to land?
Well, that's the final provocative thought maybe we can leave you with today. Just consider the additional selective pressures that terrestrial organisms face. Moving on to land introduces huge challenges like dehydration stress, different needs for regulating temperature, and just the constant physical demands of fighting gravity. How must those stressors further shape and customize the post-metamorphic metabolism, perhaps in unique ways that we haven't even fully explored yet?
A really great question for you to mull over. We certainly hope this deep dive gave you a fast track to understanding the exquisite energy management required for one of nature's most spectacular biological transformations. Thank you so much for joining us. Don't forget to like and subscribe, and please do share this deep dive with anyone who loves biology and efficient energy systems. This has been the Sand Science Experience.